So thank you again for inviting me. Probably you will realize that my talk is a bit different from what has been discussed so far. But I hope at the end it will be clear that one of the topics of this workshop about energy and the energy constraints under which neurons work is very relevant for my take-home message, that is that sleep is the price for plasticity, for the ability of the brain during wake to learn and to be connected to the external world. So this work is done in collaboration with Giulio Tononi in Wisconsin. It's funded by NIH and by the Department of Defense, and I have no conflict of interest to declare. So we are trying to understand why we need to sleep, and we spend on average a third of our life asleep, and why all animal species that have been carefully studied so far do the same. And here you see just some examples. Now, you can say that this is a trivial question, meaning we all know why we need to sleep. If we don't, we feel awful, and everything is extremely difficult to achieve. And indeed, there is really compelling evidence that sleep is needed to maintain proper cognitive function. And here you see examples of the specific aspects of function that are affected.
Excuse me, we're still seeing your title slide. So
oh,
I don't think we're seeing examples.
Maybe you can put the presentation mode on. Can you find the function for
sharing
presentation?
Maybe
usually
stop
it's
sharing.
bottom right.
Yeah, stop sharing.
Should I share again?
Make sure to share from the presentation mode. Yeah, now it's good.
Yeah, that's good.
Yeah,
now
better.
you see sleep is universal?
Yes.
Okay, sorry, but I think you could. Okay, let me see if I can move now. You see next slide?
Yeah.
Yes.
Yes, so feeling very bad about sleep loss. And here you see how at least some of the major functions that are affected by sleep deprivation, from attention to the ability to learn, to memory consolidation, but even very high level functions, humor appreciation, for instance, or risk-taking assessment of risk-taking behaviors, that is impaired by sleep deprivation. Now, and these are some examples that I want to pinpoint about specific studies that have shown that once you have this impairment due to sleep deprivation, it only through sleep that you can recover from this impairment. Just quiet wake in humans, let's say with closed eyes, will not be enough. And so we are back of why then the brain needs sleep for proper functioning. And we think to try to address this question, you have to start from the fundamental definition of sleep and the fundamental feature that distinguishes sleep from any other behavioral state, including quiet wake. And that feature is partial sensorimotor disconnection, as depicted here in this beautiful painting by Rousseau. So when we are asleep, by definition, we lose the ability to promptly respond to a potentially very dangerous stimulus. So there must be at least one fundamental reason, one fundamental function that can be carried out much better, if not only, much better during sleep than during wake, to justify why we go into this state, potentially dangerous state, for such a long period during our life. And the same is true for all animals that have been studied so far. Otherwise, evolution, we think, would have found a way to carry out the same function during wake, including quiet wake. And we think that function is related to maintaining a balance in synaptic weight, maintaining synaptic homeostasis. Now, of course, you know that synapses are the basis of how the brain works. And most synapses, at least in the mammalian brain, look like these ones. So they are axospinous synapses. So these are the excitatory glutamatergic synapses that make up maybe 80% or so of all synapses in the brain. And on the left here, you see a 2D electron microscopy rendition of one of these synapses. Now, of course, there are many of them. Just even, just in human neocortex, 100 trillion of them. And we know that synaptic activity accounts for the bulk of the brain's energy budgeted. So maintaining synapses under control is fundamental to control the ATP consumption in the brain. Now, here is the take-home message of this hypothesis that we have been testing over the last 20 years, actually. And I don't have the time to go through the details of the rationale, but this has been explained in detail again in several reviews. So basically, the idea is that when we are awake in the 16 hours or so of our wake day, we are always learning something. So what we call the ongoing learning, because by definition, we are exposed to the environment. And so we need to adapt to an ever-changing environment, and we learn. And so it's not just when we go to a lecture, to a class, that plasticity happens in the brain circuits. And the end result of this process is, at the end of the wake day, a net increase in synaptic strength, which is indicated here by these red lines in the sagittal brain. Then sleep is needed to rebalance the overall synaptic weight so that the next day we are ready again to learn. Now, this process of renormalization, we call it a smart down selection, because it is certainly, for the evidence that we have, broad and comprehensive, but still has to be selective to account for the behavioral beneficial effects of sleep. So it cannot be just an aspecific downscaling, because again, sleep, we know behaviorally is able to help our learning, memory, but it also helps forgetting, and it helps to integrate the new learning with our previous body of knowledge, and does what is called gist extraction. Now, I don't have time to go through the details of this, but certainly we have now electrophysiological and molecular potential mechanisms that could explain this process of selective downscaling. So here are the bases of what of the rationale for this hypothesis, meaning learning must happen during wake. It happens primarily, although not exclusively, but certainly primarily through strengthening of connections, which creates the need for renormalization. And this renormalization should mainly happen during sleep. And we think it's exactly because of this process that then we can account for the beneficial effects of sleep at the system level, as well as, of course, at the cellular level. Because if you think about, if you bring down overall synaptic weights, that saves the brain a lot of energy needed to sustain this activity. Now, the first points about learning being through mainly potentiation and the need for renormalization, I don't think is very controversial. And, there are many, many studies showing in neural networks that indeed, if you implement learning rules, you also need to implement renormalization rules. Otherwise, you very quickly go into saturation and runaway potentiation. But I think the fundamental difference here in our hypothesis is that many people, I'm not sure most, but many people still think that the brain is smart enough that it can learn through strengthening of some connections, and basically at the same time, simultaneously, it can actually renormalized, depress other connections so that this balance overall of synaptic efficacies is maintained at any given time. While we think, and this is basically the take-home message, that the balance is present, but over a longer time scale, which is at least 24 hours. So there is a time, the wake period, during which actually there is a net increase in synaptic strength because I am exposed to the environment. I see all these, what happened to me this morning before joining the workshop. There are many memory traces that are laid down temporarily. My brain is in a state of high arousal systems activity, noradrenergic, dopaminergic, orexinergic, cholinergic, histaminergic. They are all at higher level than during sleep, and that promotes overall synaptic strengthening. Then there is instead a time for renormalization because, and that's key, again, we go back to the fundamental feature of sleep, we are sensory disconnected from the world, so we are no longer slave of the here and how. We have an arousal system that is in low activity, which is promoting depression rather than potentiation. But very crucially, and that's probably the most important lesson that we sleep researchers have learned in 1 century or more of research, is that actually the brain is very active in a specific way. More or less, we're back to the slow waves in a mammalian brain simultaneously, and that allows the brain to comprehensively sample these synaptic weights and then produce this smart down selection. And again, if there is time in the discussion, I can pinpoint to specific mechanisms through which this could happen. But I want to give you now some evidence, discuss some evidence of the main claim of this idea that indeed there is an imbalance, net increase in synaptic strength during wake, that then get fixed because there is a net decrease in synaptic weight during sleep. And that's the evidence that we have been trying to collect over many years in different species, because we think that if this is really a fundamental function of sleep, it should be shared across animal species, literally from flies to humans. Some measures of synaptic strength are better than others, and that's our approach has been to try to use them all, basically. So some, of course, are more indirect, and I start from electrophysiological markers. Of course, you can use an evoke response and measure the amplitude or the slope of this response. But as also Mavi, I think, alluded to, of course, changes in this slope could be accounted for completely by changes in intrinsic excitability or in the excitation inhibition balance. But the evidence so far in any case is consistent with also an overall imbalance, increase in synaptic strength during wake and decrease during sleep. For instance, you can use in humans TMS and measure the slope of these responses. You can see that it goes up with time spent awake, and then it goes down with sleep. Now, you can do the same in animals with electrical stimulation, for instance. More directly electrophysiologically, you can use minis in slice, which we have done. This was our original study in mice and rats. After several hours, if you take a slice of the cortex, frontal cortex, after several hours of sleep or sleep deprivation or spontaneous wake, and you basically see that both the amplitude and the frequency of minis increases with wake and decreases with sleep. And these are just to show you very recent studies done by 3 other independent groups, basically reaching the same conclusion for minis in 3 different mouse cortical areas. Now, you can also, of course, use, and this is more direct evidence, molecular markers. And the best, certainly in mammals, is the expression of the AMPA receptors. The total amount of the GLUE1, GLUE2 AMPA receptors, so their phosphorylation level, has been shown to increase with synaptic potentiation and decrease with synaptic depression. So on the left, you see our original study in which we measure the protein levels of this receptor in synaptoneurosomes in the entire cortex or hippocampus of a rat. And we found that these parameters are basically 20, 30, 40% higher after, again, several hours of wake relative to sleep, correcting for circadian time. And the right is a more recent study by Rico Ugenir's lab in the mouse forebrain, reaching the same conclusion. So I think at the molecular level, the evidence is quite strong. Now, over the last 6-7 years, we have also actually thought that going to the ultrastructure and measuring the size of synapses was one way to very stringently test the hypothesis, because stronger synapses are also on average bigger. So if this hypothesis is true, it should be the case that at the end of the wake day, on average, synapses should be bigger and they should shrink after sleep. And so we have, in the lab, we have a serial block phase scanning electron microscope that we use. It works very well. It can acquire automatically stacks of images, in this case of the mouse cortex or hippocampus, that then, however, still need to be actually segmented manually. And that's why this is so time consuming. So, you know, we reconstruct the pieces of dendrites, the axons, and then what we really measure is this area, the axon-spine interface, which is the direct area between the axonal button and the spine head. And that's the ultrastructural measure, established measure of synaptic strength. And so basically, to make a very long story short, we have so far studied 2 areas, the mouse primary motor cortex, the sensory cortex, superficial layers, as well as the CA1 region of the hippocampus in the stratum radiatum. So these are the CA3 to CA1 synapses. In both cases, we see that on average, this ASI size decreases with sleep. This is the original study in cortex, in which overall the sleep group has the ASI size 18% lower than the 2-wake group of sleep deprivation during the day or spontaneous wake during the night. And as expected, these synapses have a log normal distribution, so most of them are small or medium. And what we see is that this decrease with sleep happens in the great majority, 80% or so of all synapses. It seems to spare the very large ones. And we don't know why, but we speculate that maybe those are the ones more strongly linked to strong established memories. And those memories do not easily change just across the sleep-wake cycle. Now, this is just to say that we recently also looked during development, because in theory, you know, when the brain is growing, there is much plasticity, and we know a lot of sleep. And so if this need for renormalization is related to plasticity, it should be true also in very young brains. And indeed, this is the case. So far, we have these are 2 we called PUPs, which is as early as we can get in mice and still being able to really define sleep and wake behaviorally. And what we see is that indeed there is a decrease in ASI size in motor cortex. Actually, it's even larger, 34% than in the adult brain, and it seems to happen across all synapses. Now, I want to finish by giving you this new study that is still unpublished, but because I think it gives an example of how this process of renormalization may indeed explain both benefits at the cellular level and at the system level for sleep. So this is a repeat into photon imaging in which mice are imaged in motor cortex superficial layers, first before and after normal sleep, baseline condition. Then the next day, at the beginning of the day, we do training in a complex wheel task that we know is sleep dependent. Then we image, and then we split the mice into groups. Half are allowed to sleep and the other half are sleep deprived. Then we image it again, and then also we test again for the task the next day. So the beauty of this method, of course, is that you can follow single synapses and see what happens to them before or after sleep or after learning. And we are using this marker SEP-GLUE1, which is, again, a marker of expression of the AMPA receptors, and is specifically this variant can only be detected when the receptors are on the surface of the synapse. And so we are really sure that they are functionally relevant. Now, this complex wheel task is, as I mentioned, sleep dependent. So the animals in 1 hour, in a session, they can learn to stay on the wheel despite the wheel moving at faster rates. Then, depending on whether immediately after they can sleep or not, the next day, the animals that were allowed to sleep started the same level of performance as the day before. Instead, the sleep deprivation, sleep-deprived animals have degraded the performance. So sleep promotes the offline consolidation of this task. We have repeated this already 2-3 times. So here is the result. When we follow these 12 animals, these are all the 1,500 or so synapses that we can follow in these animals across all these time points. First of all, you see here that after, in baseline condition after sleep, the overall expression of this GLUE1, this is the normalized difference in the expression, goes down. So this is very much consistent with what I showed you before with the other markers. Then after training here, is the ND is positive. So there is a net increase in GLUE1 expression, so in synaptic strength. As expected, there are a lot of studies showing that this skill learning produces synaptic potentiation in motor cortex. But then what counts for us most importantly is this part. So even after, immediately after learning, when if you look in the motor cortex engaged in learning, after 7 hours of sleep, the net effect of sleep is a decrease in synaptic strength. And this does not happen if the animal is sleep deprived. So it's not just passage of time. You really need the sleep to produce this downregulation. But this method, of course, allows us also, this is the overall behavior of all spines together. But we wanted to ask specifically what happens to what we call the max spines. So we are focusing here at time 0, immediately after learning, to the spines, the synapses that have shown the largest increase, like this one. So they have been potentiated the most with learning. And we ask them what happens after that. And here is what happens. So this is the time 0, where we selected the max spine as being potentiated. And you see that even 24 hours later, they are still potentiated. So they're still much stronger than before learning. But you also see, because I'm plotting here the 2 groups separately, that actually sleep doesn't seem to do anything special to the max spines per se, relative to sleep deprivation. They all lose a little bit of GLUE1 at the beginning, and then they stay there. But the difference is in all the other spines, because these max spines are only a small minority, 13–40% of all the spines. 85% are all the other spines. And that's where you see the difference. That's where you see that the majority of spines go down in synaptic strength, and instead with sleep deprivation, they don't. And so when you now express the max spine, their expression, you subtract, you calculate the difference between these two, you find a significant interaction, meaning the max spine in the sleeping animals, although in absolute terms, are not behaving differently than in the sleep-deprived animals, in relative terms, at the end of the 7 hours of sleep, have an advantage, are in a better position, simply because everybody else went down with sleep, but not with sleep deprivation. And we also find that this going down, this synaptic down regulation, so having a negative ND, is correlated with the performance. So basically the next day, the animals that are better performance are the ones that slept and the one in which the other spines went down. So I think this is our first really attempt at the single synapse level to try to understand why sleep can do it all. It can provide all these benefits. You end up after sleep with less synaptic strength, so you save energy and supplies. Of course, you avoid saturation. And also, however, you are better in this specific case, in this performance. And it seems, based at least on this data in this task, that you are better because there is an increase in signal-to-noise ratio. But this is not because the signal, the max spine, are potentiated, are increased, but because the noise, the other spines, are decreased. And I stop here, and again, I will be very happy to discuss about specific mechanisms if you are interested.